Hello everyone. In this video, I would like to look at the real-life virology of the forced evolutionary virus from the Fallout universe, or the FEV. Now, when I say real life, I mean using real-life virology principles and definitions, can we determine what type of virus the FEV is? What type of genome does it have? Phenotypes, tropism, reproduction, infectivity, and other attributes this virus might have? Well, in this video, I hope to find out. Before we get into the science, let's give a brief history on how the FEV came into being. In the beginning, the initial research that would eventually lead to the creation of the FEV started with the Pan-Immunity Virion project by the West Tech Company in 2075. At first, this was part of a larger effort by the United States government to create a cure to the new plague, a Chinese bioweapon. Through experimentation and research, they found that the virus could be used to induce genetic mutation. Over time, the West Tech and US governments would experiment with this virus and would lead to the creation of what would be called the forced evolutionary virus. According to the wiki, the FEV is a virus that is transmissible through direct contact or injection. It cannot infect through airborne transmission from an already infected individual. Through a specific phenotype, it is immune to radiation. The virus copies DNA patterns, storing them in exons and combining them with FEV. The virus, along with the induced FEV code, are then merged, which will induce mutation through the translation of the exon FEV code. This is done extracellularly, apparently, and then reintroduced back into the cell and its subsequent genome so that it can be translated later on. The severity of the effects of the virus are dictated through its dosage amount, with the current super mutants being a case of a severe overdose. Another important note is that the virus does not replicate within the host cells. Successful infection relies on dosage amount and successful integration into the host genome. There also seems to be a genetic component where individuals who do not react favorably will not mutate in a positive way, and the resulting infection will end in death. So now let's talk about the effects of the FEV. The effects of the FEV are varied and still not fully understood. The effects of the infection depend on not only the amount used, but the pre-programmed genetic code that will be used to not only induce mutation, but shape the organism. For simplicity, I will focus on the supermutant mutations for now. The specific mutagenic properties that supermutants develop is as follows. Skin color becomes a dark shade of green, or blue if nighttime, increased muscle mass and strength, skin defects and warts, loss of sex organs, and their intelligence will either be higher or lower depending on the effects of radiation pre-mutation. Now let's talk about what real-life viruses, bacteria, and other microbes which could be most similar to the forced evolutionary virus. First off, I can't not mention this, so I will here. The name forced evolutionary virus is probably the most incorrect name in all of science fiction. It is actually incredible how bad it is. Why is the name so bad? Evolutionary. Evolutionary is incorrect. In fact, it is the most incorrect. I'm not assuming many of you watching know the difference between mutation and evolution, so I will explain. Both mutation and evolution involves changes in genotype, which can lead to changes in phenotype as well. But the main difference is offspring. For evolution to occur, mutation must occur, and these changes must be passed on to viable offspring. That means fertile offspring. This is evolution. Obviously, supermutants are sterile and cannot produce any type of offspring. And so a better name for the FEV would be the forced mutagenic virus. Again, kind of important as this is literally not evolution, it is mutation, but anyways, I just had to mention it. Now let's talk about viruses. So the main takeaway from what was mentioned before was that this virus is special in that it does not replicate. This contrasts to every virus that exists in the real world. I'm not going to get into whether viruses are living or not important for this video, but viruses do replicate and use genetic material to create and proliferate themselves. There are 2 points of note here. Number 1, the virus induces mutation, which then causes the decay of the virion particles, which then leaves the cell population intact as there is no infection, reproduction, escape sequence. In all living cases, the virus infects the cell, reproduces, then either lyses the cell, which basically means blowing up the cell, or buds out through the membrane. In either case, the cell dies, the mature virus spreads to other neighboring uninfected cells. The second point, since it does not leave the cell, the closest analog in the real world that I can think of is a retrovirus. A retrovirus is well known in that it will integrate into the host cell genome where it will stay until the host organism dies. A well-known retrovirus is the human immunodeficiency virus, or HIV for short, which will integrate into the host cell, basically making it undetectable not only by the human immune system, but modern medical science as well. Now, the main difference, of course, is that in the real world, the virus does this to proliferate and spread itself to other cells. The inserted viral genome is used to make more viral particles and spread. However, the main issue is the specific DNA sequence part. As far as I know, there are no viruses that insert a specific DNA other than their own genome, as the FEV is modified based on the host cell genome. The best real analog that I can think of is CRISPR, which is short for clustered regularly interspaced short palindromic repeats. Yeah, you don't need to remember that. But this new technology isn't really new or man-made, just modified. Basically, this process is part of a simple immune system that bacteria developed to fight off bacteriophages, or viruses that infect bacteria, and with a bit of tinkering, can target specific sequences of DNA and remove them completely, or even replace sequences with completely new DNA sequences. Now, as mentioned before, I used the wiki to research this video. It is interesting that the FEV DNA and the cell DNA are spliced outside of the cell. I mention this because there are no known viruses that will maneuver outside of the cell during its infection process, at least as far as I know. This could just be author included in the wiki, I'm not 100% sure, but it does complicate things a decent amount since, as mentioned, a virus's main focus is to enter the cell, infect the cell, leverage its machinery, make more, and then leave. The only time the virus leaves the cell is once it's a mature viral particle. So once again, this sounds more like the CRISPR system that I just mentioned than a natural virus, but since this is fiction, we can just merge these two things together with no real hassle. Now let's talk about the viral genome. The genome of the FEV is actually not nearly as important as I originally thought when planning this video. Since the genome is developed in vitro, which basically just means synthetically created in a lab and not based on any natural virus, or at least any real quote-unquote virus, the actual viral genome does not have any physiological implications. In the real world, the viral genome has a significant impact on viral genome mutation, where single-stranded RNA genomes are the most unstable, leading to more mutations, whereas double-stranded DNA genomes are the most stable. I'm not getting into the biochemistry here as it is outside the scope of this video, but HIV, as mentioned, is one of, if not the most mutagenic viruses known. Fun fact, HIV can mutate around 2% of its genome in days, in one host. That might not sound like much, but compare that to humans that took millions of years to achieve the same feat to distinguish ourselves from our primate brothers and sisters. Yet, this does not necessarily mean an increase in host cell mutation. A virus that does induce significant mutation is HPV, or the human papillomavirus, which contains a double-stranded DNA genome, which is the most stable. However, it does induce significant mutagenic risk in human cells, leading to a significant increase in cancer risk if infected. So, for the sake of this video, and to reduce the amount of steps required, we could just say that the viral genome of the FEV is a double-stranded DNA molecule. Now for the most quote-unquote fictional part of the virus, tropism. Tropism is essentially the habitat of a virus. In other words, what cells do the virus infect? Does it infect skin cells, lung cells, neurons, intestinal cells, etc.? Now, from the phenotypic effects we observe and the overall description of the virus, it does seem as though the FEV infects all human cells. This is basically not possible naturally, as the size of the virus would be possibly larger than the cell it's infecting, as the sheer amount of binding proteins necessary to bind every cell in the human body would be incalculable. Of course, this is fictional, so it doesn't matter too much, but the only vector through which a virus enters a mammalian cell is through physical binding to surface proteins that coat the outer exterior of the cell, which then leads to a reaction cascade, allowing the direct entry into the cell. Since we see mutations throughout the entirety of the human body, skin, brain, organ tissue, sex organ cells, and everything else in between, there must be a mechanism through which this occurs. The most straightforward method is complete human cell infection. If I haven't mentioned this before, I will mention it now. Phenotype means physical trait. It's a biology term used to describe the physical properties that an organism contains. In the case of a virus, it can mean their viral capsule, binding proteins, DNA and RNA polymerase proteins, anti-immune system proteins, integrase proteins. Essentially, proteins provide phenotypes. It's not that simple, but for the sake of this video, we'll keep it simple. And for the most basic process of what is called the central dogma, I know, horrific name, this boils down to DNA, which is then transcribed into RNA, which is then translated into proteins. Proteins providing the phenotypic or physical differences between viruses, bacteria, and mammalian cells. This is important as we need to determine what proteins the virus itself contains pre-infection. In nature, viruses tend to contain basic proteins that jumpstart the viral production process, which then is assisted by the cell machinery. This tends to be very basic as viruses are some of the most basic organic machines in nature, but it is still important to mention. So, what should the FEV contain? First, as mentioned, we are going to need a binding protein/proteins that can bind to and infect almost every cell in the human body. Second, we need proteins that can cut out certain parts of the host cell genome along with polymerizing its own genome. There is a protein called integrase, which can be used to integrate the FEV host cell DNA into the cell genome. Finally, we would need a whole host of protein system and processes that would lead to whole organism mutation. The amount of proteins here would be significant. So for simplicity, let's just say that the specific genome contains an FEV mutation complex. Again, there's probably something I'm missing here, but it would be impossible to mention everything anyway. So to summarize, with all this information, let's end with all the features that our FEV has. The virus will have a double-stranded DNA genome, which will streamline the FEV host genome fusion process. The virus will contain no genetic information regarding reproduction, metabolism, or the viral exit process. I haven't mentioned this, but since the virus does not leave the cell post-infection, a viral envelope is not possible, so the viral particle will be non-enveloped. Through full organism tropism, the virus can and will most likely infect the majority of cell types within the human body. This is done by containing specific attachment proteins that bind to the outside of human cells, facilitating entrance into the cell. Once the virus enters the cell, it will extract a portion of the host cell genome and fuse it with its own genome. Following this, the virus utilizes an integrase protein exclusive to viruses to integrate into the host cell genome. Once infection is established, a collection of proteins we will call the FEV mutation complex will be produced that will induce severe mutations within the host cell population. The effect of these mutations lead to increased body mass and size, loss of sex organs leading to sterility, immunity to radiation, neuronal degeneration, increased aggression, and skin tone changes. And that is the end of the video. Thank you so much for watching. If you have any questions, be sure to leave them in the comments and make sure they are constructive and positive. If there's anything I missed, feel free to mention it. And if you liked the video, I would appreciate very much if you leave a like and subscribe, as it helps out the channel a ton. In the next video, we're going to be talking more broadly about how science fiction gets biology wrong. So if that sounds interesting to you, then stick around. It should be out hopefully in the next couple weeks to a month. Thanks again. I'll see you in the next video.